Hi guys, Miss Francis here today to discuss the overall processes, the big picture regarding transcription and translation. Now, transcription and translation are the two main processes that link genes to proteins, and the way I want to introduce these two processes is by comparing it to creating a new building after 9/11. It's kind of crazy to think that most, if not all of you, weren't even born when 9/11 happened, but anyhow, I'm going to tell my story. So after 9/11, Twin Towers fell and we needed a new building. So this German architect came up with blueprints and he needed to email them over to America. But the email was too big. This is synonymous with the fact that DNA is too big to leave the nucleus. It can't fit through the nuclear pores. So the architect sends a messenger, just as DNA is going to send a messenger known as messenger RNA. Well, the architect is nervous about the messenger carrying this super important blueprint. So he wants the messenger to protect the blueprint. So he gives the messenger a carrying case. Similarly, messenger RNA gets a cap and tail added to it to protect that messenger RNA as it travels out through the nuclear pore into the cytoplasm. Now, we've got our blueprints. They're in America. They're at ground zero, and we're ready to build. Well, ground zero, when we're talking about transcription, is at the ribosomes. Now, the materials such as steel and cement and the construction workers, they come in via taxi. In our case of transcription and translation, the amino acids, the transfer RNA, act as our taxi. So at the building site on the ground is a foreman, and this foreman is directing the construction workers. He's telling them how to build their building. This is provided by the letters of messenger RNA. The letters of messenger RNA are going to tell us how to build our polypeptide. So in 9/11, it's the building that's taking shape, but in translation and transcription, or transcription, I should say, and translation, we are building polypeptides. So this string of amino acids gives us the protein, which then gets sent to the Golgi body, which then gets wrapped in a vesicle and shipped out to where it's needed. So essentially, transcription and translation are kind of like a two-part movie, where part one involves transcription that takes place in the nucleus, and part two involves translation that's going to occur in the cytoplasm. Now, before we get into these processes, I would like to review the differences between RNA and DNA. RNA contains ribose as its sugar, not deoxyribose. It contains the nitrogenous base uracil, not thymine. And RNA is a single strand, whereas DNA is double-stranded. So if I were to summarize the processes of transcription and translation, I could say that transcription essentially takes the DNA language and transcribes it into RNA language in the form of messenger RNA. And this process occurs in the nucleus. If I wanted to summarize the process of translation, I could say that translation takes the messenger RNA and translates it into an amino acid sequence, which then builds our polypeptide. And this occurs at the ribosome, which we know is located in the cytoplasm. Now, the story that I told you about 9/11 involves transcription that occurs in eukaryotes. Transcription in prokaryotes is slightly different. So a prokaryotic organism such as bacteria does transcription a little bit differently. Remember, prokaryotic DNA is circular. It has no histone proteins, therefore it's considered naked. In addition, prokaryotic organisms don't have a nucleus. They have that nucleoid region. So transcription and translation can occur concurrently. They can occur at the same time. So RNA doesn't have to leave the nucleus because there is no nucleus. Therefore, there's no RNA processing. There's no cap and tail, which means there's no RNA editing. So I can use that RNA exactly as it is. It occurs in the cytoplasm and it involves the 70S ribosome, which is free. In eukaryotes, though, remember, eukaryotic DNA is a linear DNA that's wrapped around the histone proteins, where we do have a nucleus that is present, and that nucleus has nuclear pores. So inside that nucleus is where transcription is going to occur and where the RNA processing is going to occur. So what do I mean by RNA processing? I mean that the RNA gets protective ends. It gets the cap and a tail. There is an editing process that is involved in transcription. Translation now, on the other hand, occurs in the cytoplasm, more specifically at the ribosome. And those ribosomes are sized 80S. So to summarize, we call the summary the central dogma, where DNA is transcribed to RNA, which is then translated into a protein. Now, the genetic code that is used in order to determine the amino acid that's going to be coded for is called the DNA dictionary, or at least that's what I call it, where each triplet of nucleotide bases is called a codon, and then each codon codes for an amino acid. Now, it makes sense that these codons consist of triplets of nucleotide bases and not single nucleotides or even pairs of nucleotides, because then there wouldn't be enough possible combinations to code for all 20 amino acids. So here's an overall picture of what's going on inside of our cell when transcription and translation is occurring. Here we've got our DNA molecule, and we all know that DNA is composed of hundreds to thousands of genes. So we're looking at one section of a gene, a section of DNA. The DNA has been unwound, and the parent strand is acting as the template. So that DNA strand is being transcribed into messenger RNA, where one codon is equal to 3 nitrogenous bases, which then gives us 1 amino acid. That codon is translated into our amino acid. And then it's the sequence of these amino acids that determines which protein is going to be made. So if I said we had 100 amino acids, that means that we have 100 codons. But if I were to say we have 100 amino acids, how many nitrogenous bases would we have? That's right, we would have, oh God, this is awful. We have 300 nitrogenous bases. This pen is terrible. Marshall Nuremberg is the one who determined which codon matches with which amino acid. And here's that DNA dictionary that I was talking about. So how the heck do I read this DNA dictionary? Well, I take my messenger RNA and I look at the first base. And let's say the first base is A. I'm going to be looking in this box. Now I look at the second base, and let's say the second base is U. I'm going to look at the box that combines A and U. Now let's say I'm looking at my third base, and my third base happens to be G. Well, I'm going to follow that over to where my AUG, my codon, is located, and we know that AUG is known as the start codon. It's methionine is the amino acid. Notice there are 3 different stop codons. There's a UAA, there is UAG, and there's UGA. So it's important to realize that there are multiple codons that can code for the same amino acid. So notice how phenylalanine can be coded for by those two amino acids. Notice how leucine could be coded for by those two codons. Notice how leucine could be coded for by those 6 codons. So there are more than one codon that can potentially produce the same amino acid. That ends our discussion of the big picture of transcription and translation. We will get into the details of each one of those processes in later videos.